Exploring by the Seat of Your Pants Hangout. I am your host for today, and I'm Sarah. And for those of you who may be joining us for the first time, Exploring by the Seat of Your Pants is all about bringing science, adventure, and conservation to classroom across North America. And today is day 3 of our week-long ocean celebration leading up to World Ocean Day this Friday, June 8th. And today we have a special guest. Our special guest is Anna Sofia Guerra, and she is a PhD student at the University of Santa Barbara. She's also an avid diver and photographer and illustrator. And Anna is currently studying schooling fish on coral reefs and seeing their impact on the environment that they surround. So before we get started, I'm just going to go around to each classroom and give everyone a chance to say hello. So let's go to Miss Tanya's classroom. Tanya, Mrs. Del Pino's classroom from Thunder Bay. How are you doing this morning, guys? Oops, let me just unmute your mic. How are you guys doing?
We're good. Great.
Good. We also have Mr. Rotman's grade 2s from San Antonio, Texas. Hey guys, we also have Miss Marshall's class from her grade 5s from Scarsdale. Hello everybody. And last but not least, we have Miss Lovell's classroom, a group of grade 1 and 2s from Elgin, Ontario. That's also where I'm coming out of. Hi guys. All right, so without further ado, we will have Anna take it away.
All right. Hey guys, so my name is Anna, and I'm a graduate student or a PhD student at the University of California in Santa Barbara. This is on the West Coast, and I study marine science. But before I actually tell you about what I do as a researcher, I'm going to tell you a little bit about me. So I was born in Manta Rays, Mexico, a city that is really, really far away from the ocean. It's actually completely surrounded by mountains and desert. And so when I was a kid, the thought of being a marine biologist was completely unrealistic. Like, it was just not even in my wildest dreams. But I still really love the ocean, and I would watch these ocean documentaries, and I was completely intrigued by them. And every time my family would take us to the beach, I would just spend the whole time in the water and taking apart bits of seaweed and looking for little crabs. And I was just completely intrigued by this. And it was when I was about 12 years old, we went on this beach vacation and we found this nonprofit organization that actually saved baby sea turtles. So they'd go out, and because baby sea turtles the sea turtle eggs often get eaten by other animals or by humans, this organization would go out and dig out this baby sea turtle eggs and put them in a safe space and then release all the baby sea turtles into the ocean. And for me, it was this moment of like, oh my God, like I can do this for a living. I could save baby sea turtles. And so I kind of kept going on this dream of like, I want to be a marine biologist. And I really had no idea what being a marine biologist meant. I thought I was maybe going to go save dolphins. I had no idea. I just thought I really wanted to do it. And then I kind of forgot about this as I went on through like middle school and high school, and I just started taking classes, and I knew I liked science, but I wasn't really sure what about science I liked. So when I came to the US for college, I actually started working more in like taking biotechnology and medical research and working in laboratories and doing more chemistry type of work. And I realized that while that was interesting, it really wasn't what I wanted to be doing. So I decided not to take those classes anymore, and I started looking more into other types of classes that were offered. And I found that I really, really liked the classes that dealt with animals and animal behavior and conservation and how we can keep all these animals around. And I completely fell in love with this kind of work. And the university that I was at actually has a marine biology station a research station an hour down the coast in California. So I went down there, and it was this incredible place where I could actually take classes right by the water. And so I could actually see all of the things that I was studying and touch them and just like go out, explore the ocean and actually understand what was happening. So I realized I was completely captivated by the ocean academically. I really wanted to learn everything about it. And not only that, I refound this passion for wanting to be in it all the time. So it was this weird thing where I'd, go diving in between my classes, or I'd run into a class having just gotten out of my wetsuit. And it was a really, really fun way to learn. And while I was there, I also started working in labs. So labs can do many different kinds of research. And I really just wanted to learn, what does a researcher do? How can I get involved in more of this type of work? And so while I was a college student and after I finished my college studies, I continued working on many different types of projects before I started grad school. So now I'm gonna share my screen a little bit so you can see some of the pictures of the projects that I worked on. Let me see if this works. All right, so this is just gonna get a thumbs up from Sarah here that she can see my screen. Perfect, thank you. So this is a bristle-thighed curlew. This is a shorebird. And as my honors thesis when I was in college, I was studying this species of bird. This bird nests in Alaska and spends its winters on very warm Pacific islands. And so what I would do is I had cameras set out across an island and was looking at what kind of habitat do these birds like? Where do they spend their time? Where do they eat? Because a lot of these birds, their populations were actually going down. And I wanted to figure out, well, if we want to try to save some space to keep these birds around, what kind of space should we be conserving? What kind of habitat do they need? And another project I worked on was with coconut crabs. These crabs are not like your normal crab. These are the biggest land crabs that we have that live on land. These crabs the populations are really small, mostly because they are delicious and a lot of people eat them. I have never eaten one, so I don't actually know, but this is what I hear. And these guys get huge. This one that I'm carrying was maybe about 8 pounds. These crabs are massive, and their claws are really, really sharp because they eat coconuts. And so they can actually crunch through a coconut. So what we were doing is, because a lot of these crabs are hunted in many places, or they get eaten by things like introduced, dogs or cats, we were trying to figure out what kind of habitat do they need. If you wanted to create a safe space for crabs, what do you want a place with a lot of trees? Maybe you want a place with a lot of grass. We don't really know where these crabs spend a lot of their time. So we would go out at night, mostly, sometimes during the day, but these crabs typically are nocturnal, which means they come out at night. And we would put little tags on the crabs. One tag was a GPS tag. And in the bottom right corner, you can see this a little tag stuck on it. So that's a radio tag. So we put a GPS tag that logged the location of the crab every 30 seconds. It would tell us where the crab was on the island. And then that little radio tag would allow us to find the crab later. So we'd glue these little tags on the crab, let the crab go for 5 days, and 5 days later, re-find that crab and take these tags back. And then we could get a better sense of what habitat do they need? Where should we be focusing our efforts? Another project that I worked on was on manta rays. You'll sense a trend in that I like finding out where animals go, as you'll just see as I describe more of these projects. So working with manta rays is very cool. These fish are fish, and they are beautiful animals. And we were trying to find out how often they traverse through different areas. How often do they go into lagoons or out of lagoons into the open ocean? How far do they go? So every day, my coworker, Robbie, and I would stand on this boat and look for manta rays. And it's a very long kind of day. You're just going very, very slowly for many hours on a boat. And when we did see a manta ray, one of us would swim down to the manta. And once we did that, we would do two things. One thing is we would put a little tag on it. If you see on their upper corner that picture of the manta swimming, it looks like it has a little tube attached to it. That's a tag. So every time a manta went by a specific location where we had some instruments, it would let us know, hey, manta number 3 went by at 4 p.m. today, and then again at 5 p.m. So we would know when the manta was around a certain area. And the other thing we would do is take a picture of the underside of the manta. And manta bellies are kind of like human fingerprints. They are all different, so you can actually tell individual mantas from a picture of their belly, from all the little black markings they have on it. So by doing this work, we could see, first of all, how many mantas were in this area? How many individual manta rays did we have here? And of the ones we were able to tag, we could tell where they were going. And one of the last ones I'll talk about before I talk about my PhD work is I worked on gulls out here on the Channel Islands. Most of you have probably seen seagulls. You probably know them as the birds who eat your French fries. And this is true, they do eat your French fries. And what we were doing is we were actually figuring out where they go when they're nesting, while they're nesting, where they go feed when they're nesting. So these birds nest on the Channel Islands. There's a little island chain off the coast of California, and there'll be thousands of these birds just nesting everywhere on the island. And what's interesting is that some of these birds will go hunt for food and fish for food, you know, like normal seabirds do out in the ocean. But some of them eat your French fries. So we wanted to figure out how many of these birds actually go fishing like wild birds and how many of them go hang out by dumpsters. So we would catch these birds and attach little tags to them, to their tails, and let them go for about 5 days and then catch them again to get that tag back. And this is actually really cool. So you can see Santa Barbara Island below that picture of the flying bird is down there. All the birds are nesting there. And you can see the gull, that's the red track, went out to the ocean, went fishing, then went right back to its nest to sit on the egg and switch out with its mate. And then the mate went fishing. And the gull that's in yellow went out from its nest, flew about 40 miles to Los Angeles, and actually went to this little food court that had a lot of seafood restaurants. So maybe it was also eating crab or fish, but it decided to get it from a restaurant location instead of from the ocean. And hang on, stop sharing my screen right now. Cool. So all of that work led me to where I am today, towards getting my PhD. And I work in a community ecology lab. And what this means is that we study how different species interact with the environment that they live in. So I don't actually do traditional lab work. I actually, instead of petri dishes and microscopes, I have sunglasses and scuba gear. And for my PhD, I study animal aggregations. So big groups of animals, for example, a flock of birds, or you can imagine a herd of zebra. And I study why aggregations matter. So if you think about a herd of zebra, they live in the savannah, they eat grass, they need water. And so you're thinking about the environment that these zebra need to exist. But what I like to think about is, how does the zebra actually change the environment it lives in? So would that environment look different if it didn't have 1,000 zebra eating all that grass, or keeping it really short, or if it didn't have 1,000 zebra pooping all over it? That's a big fertilizer. Or drinking all the water, or trampling a lot of the baby plants. So what happens to an environment if you remove aggregations, or if you keep them there? Are they really important to keeping an environment alive? So I study this species. Hang on, let me show this again. So I study what I like to call the zebra of the ocean. These are convict tang. They're a species of fish. And I study convicting in a place called Palmyra Atoll. It's a really, really small island. It's 1,000 miles south of Hawaii. It's 2 miles by 5 mile island. It's really small. And what's really special about this place is you can see on the sign there, it says population 7. This place has never been permanently inhabited. Palmyra has no permanent population, and so it's never been fished. So it's actually a really amazing place that allows us to see what coral reefs looked like before humans ever got there. So back to these fish. Convicting are surgeonfish. Surgeonfish are called that because they have a little teeny tiny blade on the edge of, at the end of their tail, and they call those scalpels. So these are surgeonfish. And surgeonfish are important because they eat algae. So on coral reefs, you want surgeonfish because they eat all the algae. Algae is good. Algae is natural. But if there's too much algae on a coral reef, it can actually start to kill coral. And coral is a really small animal. It's related to anemones and jellyfish. And they are the foundation. They are what make up a coral reef. They're what make a coral reef what it is because they form these big colonies. You can see on this picture, all the beige structures, those are all coral colonies. They build these hard skeletons, and they provide all the habitat for a lot of different fish and invertebrates to come in and live there. They form what makes a coral reef a coral reef. But when there's too much algae, this algae actually overgrows over the coral and kills it, and so you end up destroying all that habitat. So without all these surgeonfish and other species of fish to eat that algae, you go from having a coral reef that looks like this, with many species of fish and sharks and different species of coral, to something that looks like this, with almost no fish, a lot of the coral's dead, and it's overgrown with algae. So these fish are pretty important to have around. And what I also really like about these fish that I find really interesting is that these fish form big schools. So I'll often encounter them on the reef in a school of about 1,000 fish. But then what's interesting is that I'll be swimming and then I'll encounter one hanging on by himself, completely alone. And so my question is, well, do these fish eat different amounts of algae? Do they have a different kind of effect on the coral reef by the amount of algae they consume and affecting all the other species they interact with, depending on whether they are alone, like that little fish at the bottom, or in this huge school? Is there a difference, and does that matter? So why might it be different to a coral reef whether a fish is in a school of 1,000 or hanging out by himself? Well, because maybe you're a fish in a school and you find one patch of algae, and there's still just one patch, and now you have to share this patch of algae with like all the other fish in that school. So maybe you're actually eating a lot less. Or maybe, and like if you're a school if you're a fish by yourself, then you find that patch of algae and you get to eat all of it. Or maybe because you're in a school, if you're in a place that has a lot of predators, like a lot of sharks, you might be scared to go around wandering looking for good patches of algae if you're by yourself. And you might just stick to the tiny bits that don't really have a lot to offer. But if you're in a school, you might actually be eating a lot more algae because you're able to go out exploring because there's safety in numbers and you feel a lot safer, and just eat as much algae as you possibly can. So to figure out which of this is the answer, I would go out every day and video these fish, and I would have a GPS tracker to figure out where these fish were going, how far they were traveling, and I would take note of all their behaviors. So some of these behaviors and characteristics are things like how many predators were around them. So you see the silverfish or the more bluish fish, these fish will hang around a school and follow it for sometimes up to a couple hours. And they seem to be waiting to find a distracted fish and be able to make an easy meal out of it. And this is not something they can do with a solitary fish, because a solitary fish will see the predator and freak out and then hide, and that's the end of it. But these schools actually tolerate the presence of these predators. And so maybe having these schools around actually makes it possible for some of these predators to get some meals. And other things I was looking at was, you see those little blackfish in the video? They're not the same species. They're smaller. They'll show up again. But these fish of other species they're surgeon fish as well. So they're also eating that algae that we talked about that's important. But they might be getting the same benefits from joining the school that the actual convict tang are getting, which is their safety in numbers. So even though there's a predator around, they feel safer in this big school because they can get access to that same kind of good food patches that they can find, but not feel completely exposed. And finally, I was looking at how often these fish bite on the reef. Oh, that has sound, but looking at when you find a solitary fish or a big school of fish, how many bites do they actually take? Is that different when you're surrounded by other fish or by yourself? Because maybe if you're by yourself, you have to stop biting to look around for sharks. And if you're with others, you just, put your head down and keep eating, and you kind of trust your neighbors to tell you when there's a shark around. So that's how I spend my time. Spend my time mostly stalking fish schools, figuring out what fish do, and taking note of all of their behaviors. And that's just one of the species that I study. And now I'll pause this. And yeah, I'll take any questions.
So why don't we head to Miss Delpino's class from San Antonio, Texas. If one of you want to come up and ask a question, you guys have the floor. Anybody have a question for Miss Tanya's class? Anybody? Okay, we'll come back to you guys. We'll go to Mr. Rotman's class from San Antonio, Texas. If one of you kids want to come ask a question, you have the floor.
All right, James.
Do you put tags on all the fish that you find?
Could you hear us?
Not all of them. So he asked if I put tags on all of the fish that I find. We put tags on some of them, but these fish that I was showing you, the convict tang, are very small. They're about this big. And so we don't want to put tags on them because you don't want to hurt them accidentally. And you also don't want to stress them out. So if you stress out a fish that's that small, when there's so many predators around, so many sharks, you're actually making it a little target. And so you're basically feeding a very expensive tag directly to a shark. So we try not to tag those fish. We try to tag animals that are a little bit sturdier and can actually handle that stress, because even though we learn a lot from tagging animals, you don't want to do it too much because it does stress out the animal a little bit. So you want to keep it to the point where you tag as many to learn what you need to know, but not too many.
All right, we'll move on to Miss Marshall's classroom for grade 5s. If one of you guys want to come ask a question.
So, have you ever seen any sharks?
Yeah, I've seen a lot of sharks. All the time, actually. On this island, we have a lot of shark species, and they're everywhere and have never had a problem with any of them. They've always been very friendly.
Those big crabs, don't they pinch you?
They do if you're not careful, but we try to be very careful. We keep the scary ends facing away from us. And they're actually they're very painful pinches if you do get pinched. But luckily these crabs don't move very fast. So they're not trying to catch anything that moves very fast. They're just crunching coconuts. So you can kind of see them coming towards you slowly. So you just, move away. But yeah, we do have to be very, very careful with those because those can crush bone.
Good questions. All right, we'll move on to Miss Lovell's class, her grade 1 and 2s. If you guys want to come ask a question, I think the mic might be muted on your end, so you might have to unmute the mic there, and then we can hear you and your question just fine. There we go.
How do you tag the manta rays without hurting them?
We use a tag that sticks to the top of their skin, so it doesn't hurt them. It's not super comfortable for them, but they're okay. It's a tiny little insert that slips into them, and it's kind of like if you got an injection. So it's not the best feeling for them, but it's not gonna hurt them a lot, really.
All right, we'll jump back to Miss Tanya's class now. If you guys have a question, you have the floor. I see a hand raised. Does that mean that there's a question?
I think come to our class. Hello.
Hey, if one of your kids want to come up and ask a question to Anna, you guys have the floor now. Anyone? Anyone there? Okay, you in the sweater? Yeah, you. I mean, yes, you. You can come on up. You know.
How much animals have you tagged? That's not normal.
Of different species, we tagged about 50 birds, maybe about 30 mantas. I've tagged many fish. I can't even. Yeah. And we tagged maybe about 10 or 15 crabs.
Okay, that's a good amount of. If
Yeah, a lot of animals with little tags on them.
Okay, what about the one raising your hand? The boy in the gray long sleeve there.
How do you go on the boat every single day?
When I'm doing work in the field, I do for the most part. All of my work is on boats, typically smaller boats. And yeah, every day we have to load up the boat and get all our equipment there and bring our packed lunch and spend, you're spending about 8 hours a day on the boat every day.
Awesome. Go ahead, Kyle.
Okay.
Up there so she can see you. Yeah. Here you go. Now you're really close. Go ahead. How deep have you ever went?
Scuba diving, I've gone about 40 meters is the deepest I've gone. Maybe a little bit more than that. So, or 130 something feet, depending on which system, if you're in the US or Canada. Yeah, about 40 meters or 130 something feet. That's about the deepest I've gone.
Okay, it looks like we have time for one more question per classroom. So we'll come back to Miss Tanya's classroom and we'll go to Mr. Rotman's classroom. If you guys have one more question, the mic is muted on your end. So if you want to unmute your mic, you have the floor.
Okay, Cruz. All right, they're coming up. All right.
Have y'all seen any different sharks?
Have I seen any different sharks?
Yep.
Yeah, I typically see black tip reef sharks, white tips, gray reef sharks. There's a lot of different species, nurse sharks, lemon sharks. I've seen, yeah, across the different places that I've worked in, maybe about like 10 different species of sharks or so.
Okay.
All right, we'll go to Miss Marshall's classroom if you guys want to unmute your mic. We're all ready to hear your question.
Okay. Can you hear us now?
I should.
Good.
How many species have you saved?
Have I saved?
Yeah.
Well, none of them are extinct, so that's good. I don't know if I can measure how many I've saved. I'll say that I'm still trying to make sure they stay around. So it's hard to answer that question because we don't know. While still around, we say we're doing a good job. Is all I can say because we don't know how many are saved until they're no longer here. Then we're like, oh no, that one was wrong. So far so good. They're all here.
There you go. You're doing your job as a conservationist, by keeping them conserved. Okay, Miss Lobel's classroom.
Okay, one more question. I have a voice so oddly go up. We're coming.
No problem. Take your time.
Have you ever seen a great white shark?
I have not. No, I really want to see one, but I've never seen one yet. One day. I'm keeping my fingers crossed that I'll be diving and it won't sneak up on me, that I'll have planned it. But I do really want to see one day.
Have you just not dove in the areas where there is one?
I dive a lot in California, actually, where there's a lot of great white sharks. They're shy. They don't want to mess with humans. And so they'll hear you and they'll swim away. So, there's areas that I used to dive in where there's a bunch of great whites and we never saw one, not once. So they're not looking for us. If anything, I think they're trying to stay away.
Okay, we'll go back to Miss Tanya's classroom if you guys want to finish up with one more question for Anna. Anna, if you want to take a pic of all of the excited hands you have there.
I would love that. Hang on.
I think you've got our class mixed up with someone else's because I haven't seen it yet, but we are talking to you and we did get to ask questions. Tanja Del Pino's class is in Thunder Bay and we're at the very end of the screen picture there, but it doesn't come up when you tell us we're on.
Oh, sorry about that. Yeah, so I guess it might just be mislabeled. So
that's okay. Go ahead, take your pic.
I got it. Thank you, guys. I got the pic.
Any students you want to choose from there?
Go ahead.
How much years were you doing this?
Can you repeat that, please?
How much years were you doing this?
Years.
How many years have you been doing it?
Oh, how many years have I been doing this? I have been doing this for about 10 years now, maybe 9 years now. And hopefully many more to come.
Yeah, it takes a lot of work to get to where Anna is right now. Lots of school.
It's a lot of work, but it's possible. It's for anyone who's actually really interested in the ocean or science or conservation or animals, it's a good path. It's a good path. It's a good one. You learn a lot and you have a lot of fun.
All right. Well, thank you everybody for your questions today and for participating. Anna, is there any way that people could reach you and follow you on social media if they would be interested in learning more?
Sure, yeah. I don't know if my website is linked to that. I can definitely email you or email the list. My email, if anyone has more questions or wants to know more about being a marine biologist. And happy to share. I have an Instagram account where I post all my underwater pictures and science stories. But yeah, definitely can send you that for you to send to the classes. And if anyone has questions about what, if they're interested in this path, happy to answer them.
Amazing. So to thank Anna for joining us today, why don't we all give her a good big round of applause and say thank you for all the classes.
Thank you.
Amazing. Thank you for everyone's participation. Don't forget to celebrate World Ocean Day on Friday. We still have some more hangouts going on. So if you guys want to join in for more, we have more scientists and explorers coming in to teach you as much as we can about our oceans.
Thank you.
So thank you, everybody, and we'll be signing off.
Bye.